Okay, so welcome everyone to the population dynamics meeting of today. Today we have Katja Taute, and I will present our speaker. I will try to share my screen first of all. Let's see. I, I hope you can see the full screen. Katja Taute from University of Leipzig in Germany. The title of the talk is Bacterial Navigation in Complex Environments. Couple of words regarding the speaker of today. So she got a PhD in physics at the University of Leipzig, and then she went for a postdoc at Amolf in Amsterdam in the Netherlands, and then researched at Harvard University and professor for microbiology in Munich. And now she is professor and also PI at the University of Leipzig. So she is interested in optimality and constraint of bacterial motility strategies that she investigates by using experiments and theoretical models to understand the active and passive dynamics of bacteria in space and time. So central to her research there are a few questions, among which on how does global spatiotemporal organization emerge from single particle dynamics in active matter systems, so more general, more than bacteria. What is the role of individuality in population dynamics? And as I told you today, she will speak about bacterial navigation in complex environments. So with this one, I will stop sharing my screen and give the stage to Katja. Thank you, Katja, for being here, and you can share your screen, please.
Thank you very much for the kind introduction, Marco. Can you guys hear me okay? Can you see the screen? Yes?
Yes.
Excellent. Okay, let's get started then. All right, so I would like to share with you today a little bit of work that our lab has been doing, investigating how bacteria navigate complex environments. And I would like to start out by telling you why we care and why you should care too. So bacteria navigate a very wide range of different kinds of habitats on our planet. For example, on the left, you see a cartoon of bacteria navigating in the ocean. So you can imagine every time a fish poops, that piece of organic debris starts sinking down in the ocean. And whether it arrives at the bottom of the seafloor or ends up back in the atmosphere as carbon dioxide depends on whether somebody manages to eat it in the meantime. And we know that bacteria use their ability to swim and to navigate to find these particles and associate with them and try to eat as much of them as possible before everything ends up at the bottom of the ocean. And because of that, bacteria actually play a huge role in carbon cycles and basically in the global climate system. Bacteria navigate not just simple habitats like the ocean, they also inhabit much more complex places, like, for example, the gut. No doubt you are aware that you have more bacterial cells than human cells inside your body. A lot of those, or most of those, reside inside your gut. But not everybody there is your friend necessarily. There might also be the occasional pathogen. And for some pathogens, we know that they use the ability to swim and to navigate to make us sick, though typically we don't really know the mechanistic details that underlie this. We're not the only ones with microbiomes. Plants have microbiomes too. And the picture on the right here shows you a fluorescence image of a plant root in blue in the background, and this green fuzz around it are bacteria that associate with the plant root. This is called the rhizobiome. And a lot of plants depend on such bacteria to help ward off diseases and also to help take up nutrients. So this association between bacteria and plants is actually hugely important in agriculture for yields. And we know that a lot of such symbiotic bacteria in the soil also use the ability to swim and navigate to smell the plant roots and swim towards them. Okay, so bacteria navigate lots of different habitats, and all of these situations are quite important to us in health, disease, climate, etc. How do they do it? Now, physically or mechanically, the problem boils down to moving a microscopic object in a very viscous environment. And the solution that most bacteria have settled upon is to rotate a propeller. These propellers are called flagella. They're helical filaments that are rotated against the cell body by a rotary motor. So both the cell body and the flagellum spin in opposite directions. And because the flagellum is helical, it pushes the cell forward like a propeller or pulls it backward. So this you would think is kind of like the hydrogen atom of bacterial motility, the simplest possible system. We should know everything about this. But it turns out we actually know a lot more about a more complex system that looks like this. This is E. coli. E. coli has a random number of such flagella at random positions on the cell body. So it's mechanically really messy, but nevertheless, it is the organism that we know the most about for historical reasons. And then there's a whole zoo of different kinds of flagellar architectures out there in nature. But let's start with the one that we know best, with E. coli. E. coli motility has been studied for many decades, and we know now that they do something called run-tumble motility, which essentially means that the trajectories show more or less straight segments of more or less constant speed, where the bacterium is more or less just swimming in a line. And these are interrupted by very short reorientation events that we call tumbles. And we also know the flagella dynamics that underlie this. So these runs, these straight segments happen when all the flagella rotate in the same direction, counterclockwise, pushing the cell forward. And a tumble occurs when one or more flagella change their rotation direction. Then the bundle jams. The opposing flagellum has to peel its way out, and it can push the cell off course. And only when it's has rejoined the bundle with its original rotation direction, the cell can swim again. So in the video on the right, you saw the cell coming in from the sort of top left. Now it's being pushed off course by a flagellum that came out, and now it's leaving to the bottom left. And we also know that E. coli can do chemotaxis, that is to bias this random walk to move up chemical gradients. And the way they do it is that they measure chemical concentration as a function of time. They determine whether life is getting better or worse. And if life is getting better, then they lower the probability of these tumbles such that runs that go in a favorable direction are longer on average. And this we've understood for many decades now. But what about these other bugs out there? In particular, what about this simplest possible system here? Somewhat ironically, for a very long time, it was assumed that what this thing could do would be perfectly obvious, so nobody bothered to look very hard. So you rotate the flagellum one way and it pushes the cell forward. You rotate it the other way, and then low Reynolds number hydrodynamics tells you that everything has to be time reversible, so it should just pull the cell. So you can just go forward, backward, with only a rotational Brownian motion to randomize your orientation. And it was only less than 15 years ago that it was discovered that that is actually not what they do. They have a much more exciting pattern where not only are there two different kinds of runs, so the forward and backward runs, but there's also two different kinds of turns between which they alternate. So every second turn is a reversal, so basically a 180-degree reversal. But every other turn has more or less a right angle. This is called a run, reverse, flick pattern. So these right angle turns are called flicks. And so this was quite a surprise. But nevertheless, so E. coli can do run-tumble motility and swim around and do chemotaxis. These other bugs with a single polar flagellum, they can also do a random walk and do chemotaxis. So what difference does it make? Is all this variability in architecture just biological arbitrariness? Does it mean anything? We think yes. And the reason why we think that is that flagella are rather costly to make. So we know that if we overexpress flagella, the growth rate really, really goes down. And if we knock them out, you have a growth rate benefit. So it seems like flagella architectures are likely under selective pressure because making these flagella is so expensive. So the bacteria have a very good reason to not make more flagella than they really, really need. And one of the questions we have is, well, how many do they need? What difference does it make whether you have one architecture or another? And a convenient model system for addressing this question is a bacterium called Vibrio alginolyticus that can have two different kinds of architectures in the same species. And this is not the only bacterial species that does this. Interestingly, this is found in quite a number of species, and almost all of them are pathogens, which is very, very suggestive. And what takes the bacterium from one architecture to the other is physical stimuli. So this is what it looks like when it swims around in water. So Vibrio algeneticus is a marine pathogen and mostly makes fish sick, but it can also infect humans. And the moment it feels that the environment is something other than water, it starts to make these lateral flagella in addition. So this is a scenario of physiological adaptation to the mechanical environment. And this is a very convenient system for testing the effect of two different architectures in the same species, where presumably then everything else is comparable. And because this is an adaptation that is specific to the mechanical environment, that brings us to the working hypothesis that maybe more broadly, flagellar architectures might be evolutionary adaptations to the mechanical environment that the bacteria have to navigate. So this is what we're trying to figure out in our model system here. So, okay, so we have this bug, Vibrio algeneticus, we already know, and it's in this form with a single polar flagellum. It does something called run-reverse flick motility. But about this other form, when it has lateral flagella in addition, we don't know very much. Practically nothing. So the first thing we want to be able to do is to produce these architectures in a controlled fashion. That's relatively easy. So the two main phenotypes we're working with are the blue and the red one here. We have two strains we're working with. We have a wild type strain, which will make these lateral flagella in addition to the polar flagellum when we grow them in a viscous growth medium. And then we have another strain we call Prof only. So that one can only make the polar flagellum and not the lateral flagella. And we can grow that one under the same conditions, and it will not make lateral flagella. And then we can wash them and stick them into whatever environment we want to. And well, what environments should we look at? Vibrio algeneticus is a fish pathogen, as I already mentioned. It can also make humans sick as an opportunistic pathogen, and it's not exactly picky in what habitat it picks. So it can cause gastrointestinal infections, wound infections, eye infections. So that's probably very diverse environments. But what a lot of these organic biological environments have in common is that their mechanics are probably dominated by large polymers. So we selected polymer solutions and polymer hydrogels as two sort of categories of model systems to look at in the lab. Not in the expectation that we exactly mimic the natural environment, but more in the hope that we capture some general features which might then hopefully also apply to a broader range. And to look at them then, we use a high throughput 3D bacterial tracking method that I developed during my postdoc days. So here we're looking at bacteria swimming around just using phase contrast microscopy. And we use the diffraction rings that appear when the bacteria move away from the focal plane to assign a vertical position. And the way we do this is that we compare the images of the swimming bacterium against a reference library that shows us what these patterns look like at what vertical position. And we always find the best match computationally and thereby assign the vertical position. And then we do that for all the frames to piece together a trajectory. And we do that for all the bacteria in the field of view, which is hundreds of microns in each direction. And the playback rate of the movie I'm showing you here matches the recording rate of the original video. So you get a sense of how quickly we pile up data. So we typically have about 100 bacteria in the field of view at a time. And within minutes, because bacteria swim in and out of the field of view, we gather thousands of trajectories, which we can then analyze. Now we want to do this in complex environments. That can get a little bit messy. This is what it looks like when we use phase contrast microscopy to look inside an agar hydrogel. It looks optically hopeless at first, but that background is static, so we can correct for it. We can just subtract it out. And then we are left with all the bacterial patterns, and we can do 3D tracking as usual. Okay, now we have the bacteria, we can see them moving around in different kinds of environments, but if we want to make some statement about what is good and what is not so good, we need a metric. And the metric that we choose is chemotactic performance. So we give the bacteria the task to climb a chemical gradient, and we ask how good are they at that task in these different kinds of environments. And this essay was developed by a very talented postdoc who's now a junior professor in Aachen, Marianne Grognon. What we did here is to use an off-the-shelf microfluidic device where you can have two large reservoirs of different chemical concentration. So on one side, so this yellow stuff here is something that the bacteria like, a so-called chemoattractant that they will swim towards. And then we have a uniform concentration of bacteria throughout the device. And here we have a narrow channel that connects our two chambers of different chemical concentrations, such that we end up with a linear concentration gradient in the middle. In the bottom of the chamber is glass, we can see inside, and watch all the bacteria navigate in 3D in that gradient. And then we again get these massive three-dimensional trajectory data sets where we can do really nice population level statistics. In particular, we're interested in the chemotactic drift velocity. So that's the average speed of climbing the gradient, basically a performance measure for chemotaxis. But at the same time, we retain access to the full three-dimensional behavioral information for every single individual, and they're potentially all different from each other. And having these two levels, the individual level and the population level, just from the virtue of having these massive data sets, allows us to connect these two levels together and gain some mechanistic understanding. So basically to understand what kind of performance arises from what kind of behavioral traits. Now we're doing this in different environments, either in just buffer solutions, so practically water, in polymer solutions, or we put a hydrogel just where the gradient is. And we'll start with the first case where we basically just have water and we ask, what is the chemotactic performance of these two flagellar phenotypes that I just introduced? One that has only the polar flagellum and the other one that has the lateral flagella in addition. And we're plotting the chemotactic performance in terms of the chemotactic drift velocity, so the speed of climbing the gradient, as a function of the gradient strength. And what you can see here is that regardless of what the gradient strength is, it's always better to only have the polar flagellum and not the lateral flagella in water. And we could also look at controls to convince ourselves that the cause of this really is the lateral flagella. So basically, if we take the wild type and we grow it in a medium that is not viscous, it makes a lot less lateral flagella, and then you recover behavior that looks basically like the polar-only mutant. So the lateral flagella do really cause a decreased performance in water. And of course, we want to know why. The answer turns out to be relatively simple. If you have lateral flagella, you swim more slowly in water. So it seems that having these lateral flagella, at least in water, causes more drag than thrust. So it makes you swim slower, and swimming speed is perfectly sufficient to explain the differences in chemotactic performance that we see between the two strains or the two phenotypes. Okay, so in water, having these lateral flagella is no good. They make you swim slower and as a result also reduce your chemotactic performance. But what about these more complex environments? So here, what we are plotting on the vertical axis is again chemotactic performance. And what we have on the horizontal axis is some kind of concentration measure. So for a polymer solution, we have the bulk viscosity as a metric. And for the hydrogel, we have the concentration of agar. So agar is just the hydrogel that we use as an easy lab example here. And in both cases, what you can see is that while you start out in the basically water situation where the red guys do better, so the ones with only polar flagella do better, as you increase the concentration, there's a threshold value above which it is better to have lateral flagella. And this is true in both cases, both in the polymer solution and in the agar hydrogel. And of course, again, we want to know why. And we start out looking into our polymer solutions. What we qualitatively see in terms of trajectories is very similar behavior compared to before, only much slower, kind of natural, right? The environment is very viscous, so the swimming speeds are much lower, but they're still doing non-reverse flick motility. And it turns out, so we investigated many different polymer solutions to find out whether viscosity is a key driving force here. But it turns out that the upshot of all of these very careful investigations basically was that the main factor, again, that determines chemotactic performance is the swimming speed. So here we are collapsing different concentrations of many different kinds of polymers. And I was plotting chemotactic performance versus swimming speed. And all of these data seem to fall roughly along one big trend line. So the conclusion here then is that in these complex polymer solutions, it is advantageous to have lateral flagella if they make you swim faster. So again, swimming speed is the main thing that explains differences in chemotactic performance. In water, having only a polar flagella makes you go faster. And in this complex polymer solution, having lateral flagella in addition makes you go faster. But what about these hydrogels? So here, what we see is that the trajectories look qualitatively different in the sense that the bacteria spend a pretty large fraction of their time just being stuck, presumably in pores inside the hydrogel. So they're swimming a little bit, they're wiggling around in one place, and they're swimming again and wiggling around in one place. So we presume that they're being stuck, that they're getting stuck in the hydrogel, but we don't really know because we don't see the hydrogel. So we're somewhat conservatively calling this stalling in the sense that we just see the bug is slowed down for an extended period of time. So what we're doing now is we're not worrying about running and turning or anything like that. For now, we're just saying, is the bug swimming or is it stalled? And that's an example time series you see on the right here, where the pink intervals are the phases where the bacterium appears to be stalled. What we saw in the other environments was that swimming speed was the main determinant of chemotactic performance. So you may wonder, when the bacterium is swimming, do the lateral flagella make it go faster? And the answer is a resounding no. So here we have the hydrogel concentration on the horizontal axis. And in the top plot, we are plotting the swimming speed during only the swimming segments. So we throw everything away where the bacterium is stalled, and we only ask when it is swimming, how fast are they swimming? And here, what you see is that if you have lateral flagella, you're basically always slower than if you have polar flagella. So the situation is very similar to water, where having lateral flagella actually makes you slower. And if we look at chemotactic performance for only the swim segments, so again, we throw away the phases where the bacterium is stalled. We only look at time points where the bacterium is swimming. Then the advantage in chemotactic performance of lateral flagella goes away. So whatever benefit lateral flagella give you for navigation in hydrogels must have to do with the stalling events rather than with the swimming events. So it's worth investigating the statistics of stalling. In the top plot, we are looking at the fraction of time that the bacterium is stalled for 2 exemplary agar concentrations. And what you can see is that if you have lateral flagella, you spend a lot less time being stalled than if you have only a polar flagellum. Now that could be either because you can free yourself more easily from a trap, or it could be because you don't get trapped so frequently to begin with. So we can pull out the stall durations and the stall frequencies. And we see an effect for both of them, though maybe somewhat more clearly for the stall frequency. So somehow, if you have these lateral flagella, you don't get trapped so frequently. But we also found that you're swimming more slowly. So maybe you're getting trapped in the same places. You just take longer to get there. So to address that question, we pull out essentially a mean-free path of travel between stalling events. So we ask, in one swimming phase, how far does the bacterium travel from beginning to end? And we call that a mean-free path. And we want to compare that between the two flagellar phenotypes. But of course, we don't want to compare apples to pears. So if we make this comparison for bacteria moving at very different swimming speeds, it would be unfair. So we first sort the population by swimming speed, and then we compute how far do you get in a swim phase. And what we see then is that lateral flagella help you to go further inside the gel, almost regardless of swimming speed. So somehow with the lateral flagella, you're avoiding getting trapped. Of course, we wanted to know whether the lateral flagella have an active or passive role in this. So for that, we can use the fact that the motors that drive the two kinds of flagella are different. They're driven by different fuels. So what we can do is we can poison one of them selectively. So this CCCP basically poisons the fuel for the lateral flagella. And what we see is if we if we basically stall the lateral flagella so they don't rotate actively anymore, then the stall fraction of the bugs with the lateral flagella now matches that of the ones with only polar flagella. And the benefit in chemotactic performance also disappears. So lateral flagella do seem to play an active role in navigating these hydrogels. To summarize that part of the talk so far, we have investigated chemotactic performance for two different flagellar architectures in three different types of environments. And what we found is that in water, it's better to only have a polar flagellum because that helps you swim faster. In some complex polymer solutions, having these lateral flagella actually makes you go faster. So, and that gives you then a better chemotactic performance. And in hydrogels, having lateral flagella is also advantageous, but for a completely different reason. And this is something that totally surprised me. So I expected that we would find some universal principle by which lateral flagella is helpful. Lateral flagella are helpful. But instead, we find that in one situation, they are helpful because they increase the swimming speed. And in the other situation, inside the hydrogels, they do not make you go faster at all. They actually make the bacteria slower, but they're still helpful because they prevent the bacteria from getting trapped inside the hydrogel. And the way we think about this is that, well, if you have a single polar flagellum, you can basically just go forward and backward. And if the way out of the pore is towards the side, then you might have a hard time finding it. Whereas if you have these lateral flagella, which can only push, but you have many of them attached in different positions on the cell body, you might have a lot of degrees of freedom that allow you to move around laterally and try out different directions for escaping from the pore or not getting trapped in the first instance. Now, since this is a population dynamics seminar, I want to also discuss a little bit how this what outcomes you might expect at the population dynamics level. Now we have only evaluated essentially the navigational benefits of having different kinds of flagellar architectures, but in the real world, you have to pay for everything you get. And what we can see is that if you want to make these lateral flagella, it costs. So similar to what we saw before for E. coli flagella, the more lateral flagella you make, the more of a growth rate cost you pay. So here, on the horizontal axis is basically the viscosity of the growth medium. And we know that the more viscous the growth medium is, the more lateral flagella they make. And we see that this corresponds with an increase in the doubling time. So they pay in terms of growth rate for making these lateral flagella. So then you can wonder, how does this play out in a real life scenario where your success depends both on growing fast and on navigating fast? And a beautiful assay that involves both are these so-called soft agar assays, where you essentially have chemotactic range expansions. So this is a Petri dish where you start out with a droplet of bacteria put in the middle. The gel is so soft that the bacteria can swim it, basically like the gels that we've also investigated here. So also agar hydrogels with a nutrient in it. And what you get that is in time, this population expands, and you have sort of a traveling wave at the atrium. This is a population of bacteria that performs chemotaxis on a gradient that they are creating themselves by consumption as they move along. So a very beautiful assay that relies both on being able to navigate really well and on the ability to grow. And what we did then is we did this assay with the two different strains we had, so the wild type strain and the one that cannot make lateral flagella, and ask how fast can they conquer the territory. And that is what is plotted here. So on the vertical axis is the expansion rate. So basically some kind of measure of success of colonizing the territory. And on the horizontal axis is concentration. And the red data are again for that mutant that cannot make lateral flagella. And the blue data are for the wild type, which can make as much lateral flagella as it wants to. It can, in principle, adjust to the conditions that are present. So you might think that perhaps the wild type should be able to optimize its expression of lateral flagella to the environment. And what we see then is that at high concentrations of agar, so in relatively dense environments, the wild type does indeed do better than the mutant that cannot make lateral flagella. But there are also concentrations that are more dilute, where the mutant actually outperforms the wild type. So what the wild type does, the choice that the wild type makes in terms of expressing these lateral flagella, is not necessarily optimal for all environments. And in the context of this trade-off between being able to navigate really well and being able to grow really well, I also would like to comment a little bit on a prior project that we started at Amov a long time ago and then later completed at Harvard, where we investigated, well, what if you have two species that differ in how they navigate this trade-off between growing and motility and how they assign resources to one or the other. And this is something that you actually do see in natural populations, that there really is a trade-off. If you look at bacteria taken from the same environment, so natural isolates from the same environment and the same species, they typically exhibit exactly that trade-off. So the ones that can navigate better are typically the ones that don't grow so fast. And so we did competition experiments with these two strains. If you do this in a shaken environment, that's of course totally boring. Then the one that grows faster always wins. It only becomes interesting when you do this in an environment where the spatial context matters. So what we did is we took a mixture of these two and inoculate them in one of these soft agar assays and then observed over time how they would conquer space. And to our surprise, what we saw is that within a few days, these two strains do indeed conquer the entire territory, but they segregate spontaneously from each other. So we started out with an equal mixture in the middle. And then initially, the dynamics are dominated by the fast-growing strain. So that's the red one in this picture here. But at some point, the blue one makes it to the outside, so the one that navigates better. And as soon as the blue one has sort of caged in the red one, so basically cut off the nutrient access for the red bacteria, it can take over the rest of the territory. I play this again for you. So first red, and then the blue one fences it in and captures the rest of the territory. We repeated this experiment with different initial ratios of strains. So this was for a 1-to-1 ratio. And we did a whole series where we varied the initial ratio of the two strains from 1 to 1,000 to 1,000 to 1. So basically a millionfold range in initial conditions. And somewhat surprisingly, the outcome always looked quite similar. So you always get this dot and donut pattern, quantitatively somewhat different, but qualitatively very similar, suggesting that there's some kind of stabilizing factor here that drives the system to a preferred state. To investigate that, we quantified as a function of the initial ratio of the two strains, which varied from 1 to 1,000 to 1,000 to 1, so a millionfold range. We quantified the fold change in the ratio. So how much does the final ratio how much is the final ratio of strains divided by the initial ratio of strains. So how much did the ratio change? So then if this number is smaller than 1 in this definition, then A, the red one, has gained over the blue one. And if it's larger than 1, then the blue one has gained over the red one. And so ignore the gray dots. This is for a spatially uniform environment, not a spatial contest. The interesting data here are the blue ones. And the dots are experimental data. And what we see here is that for small initial ratios, so we start out with a lot less B than A, B gains over A. So you get values larger than 1 here. And if you start out with very little of the red guys, then you get more of the red guys in time. So basically, the system drives itself towards a preferred value, a preferred ratio of these two strains, which basically means that if you repeated this assay over and over again, so you always mix up everything in the end and use that to inoculate a new plate, you would get stable coexistence of these two species. So basically in a circular or periodic competition, you get stable coexistence. And it turns out that this is relatively simple to explain on the basis of nutrient competition alone. So we drew up a relatively simple model. So it's basically a modified Keller-Siegel model. So basically like a drift diffusion model where we also have growth present in. So we're modeling the bacterial densities as well as the nutrient density as a function of time and space. We have essentially monotype growth. We have consumption. We have diffusion and chemotaxis. So the diffusion here reflects random motility. And these are basically all the ingredients that we need to predict exactly this kind of behavior. So the line here are simulation results where we basically simulated these dynamics and get out something very similar. And what is nice is that we didn't use any fit or fudge terms here. All the parameters that go into this model were determined with in separate experiments on single strains. So the growth rates we can measure independently. We can also measure these chemotaxis and the diffusive abilities in separate experiments, and we stick those into the simulations, and we get something that agrees with about an order of magnitude with the experimental values, which I thought at the time pretty impressive, given that this is such a simplified model. And the reason why this is interesting is that typically when you want to explain coexistence of species, you have to come up with some rather complicated model. So if these two species are not eating apples and pears, so if they don't have two completely different niches, and they're competing for the same resource, then typically you have to introduce some kind of asymmetric interaction to explain how they can coexist. So you can assume that they crossfeed, so they each depends on the other, or you can imagine some kind of circular warfare, like a rock, paper, scissors game to achieve coexistence. Whereas here, we don't need that. We have two species that only interact by competing for the same resource. But because they're subject to this growth motility trade-off and they navigate in space, they can still coexist. And this is just one of quite a number of studies in recent years that shows that how you move across space, or that you move across space, can really alter ecological dynamics, not just bacterial coexistence, but also the evolution of antibiotic resistances or selection for cooperative traits. So I'm going to wrap up here. In the first part, I showed you that physiological adaptation in flagellar architectures improves chemotactic performance in complex environments, which may take as evidence that perhaps flagellar architectures may also represent evolutionary adaptations to different environments. And more generally, what we learn from this is that mechanics really constrains how you can navigate. And mechanics here means both the hardware that you have available as a bacterium, as well as the mechanical properties of the environment that they're in. And how these two interact constrain how you can move about. And in the second part, I showed you that if you have a trade-off between the ability to navigate and the ability to grow, that can enable coexistence of species competing for the same resource. I would like to briefly acknowledge the people who contributed to this work. So for the first part, that was mainly driven by postdoc Marianne Grognaud with the help of a couple of undergrads. Whereas the second part, that was mainly driven by PhD student Sebastian Gude. And this is our current lab, still relatively new at the University of Leipzig. And close here, and I'll be happy to take questions.
Thank you, Katja, for the nice talk. So we have some minutes for the questions. I would like the audience to raise the hand or just open the microphone. Do you have any question at the beginning from someone? Yeah, let's start with Rosalind, please.
Yeah, thank you for a really nice talk. It was great. I have two questions actually, which are sort of two different questions. First question, maybe I'm not very well educated about hydrogels and polymer solutions, but I don't really have a sort of a clear view in my head, like when a bacteria moves through either your polymer solution or the agar, what's it actually encountering? Like, I thought the agar was actually quite dilute, and so I'm not.
Yes.
I have a good vision how it gets trapped. I guess that's what I'm saying.
Yeah, so the way we imagine this is basically large pores. So we basically imagine, well, actually, we have a figure here somewhere as well. Maybe I can go back to that. So here, this is an electron micrograph
Yeah, I see.
that shows an agar hydrogel, probably at a denser concentration than what we typically use. So for us, the pores are probably much larger. So basically, you have like fibrous polymer strands with large pores in between. And some of the bacteria are swimming through these pores. And we think that every now and then they end up stuck in one that is just a little bit too small and doesn't have an accent that's large enough for them.
So it's not like tubes, it's more like, bad luck that sometimes you end up in a little bubble or something.
That's sort of how we're imagining it, yeah. Yes.
Interesting.
Yeah, I'm thinking of it sort of like, these climbing spiders that you have at playgrounds.
Yeah, okay. Yeah, thanks. If no one else has a question, I have another question.
At the moment, no one else. You can go on.
Okay. So about the second part of the talk with your beautiful coexistence experiment, I was wondering, how does it depend on the system size? Because like you have the red ones that get trapped in the middle, and then the other ones sort of keep on going. So presumably, in a bigger dish, you would end up with a different species ratio, wouldn't you?
Yes. Yeah, that is absolutely true. So the preferred species ratio would depend on the size of the physical habitat. In a way, there's like a sort of a critical size, you could say, where the blue one manages to outrun, well, the fast swimmer manages to outrun the fast grower. And that sort of sets the size of the fast growing population. And then the fast swimmer gets the rest of the territory, however big it is.
Yeah. Okay. So if you're a fast swimmer, that's probably a good strategy in a big territory.
Exactly. Yes. So whether or not it's worth to swim depends on how large the space is that you have available for traveling in.
Oh, thanks. That's really interesting.
Any other question from the audience? It's Friday. People are sleeping.
Fair enough.
Here we are. Okay, so Maria is asking, have you looked at how variation in flagell architecture in different species correlated to environments?
Not us specifically, but what is known from the literature or more generally in the field is that there is some loose correlation between habitats and architectures in the sense that in the ocean, a lot of the species only have a single polar flagellum, whereas architectures like E. coli's architecture, like with many flagella, are things that you are more likely to find in the gut or in soil. Like E. coli has its architecture as a gut bacterium. Bacillus subtilis also has this architecture, but it's a soil bacterium. So we may speculate that maybe in general having more flagella is somehow better for more complex environments. In the past, this has mostly been considered from a perspective of how much these flagella cost. And the argument has been that the reason why the marine bacteria have only a single polar flagellum is because they can't afford anymore, because the ocean is such a nutrient-poor environment. Whereas in the gut, there's a lot of food, so you can afford more flagellum. And here what we're finding is that there may be more to the story. It might not just be the cost that differs, but also the benefits that you can obtain.
Yeah, thanks. Rosalind, please.
Yeah, sorry, that question made me think of another question. I was just thinking, you have this beautiful collection of individual trajectories in the first part of the talk. What can you say about the variability between individual bacteria? You must be able to say a lot, I guess.
Oh, yeah. Yeah, this has ballooned into a huge topic of research in the group. So you can invite me back for another talk. And I can talk about phenotypic heterogeneity and the impacts it might have at the population level. We now what we can see is that from what you see at the population level and then population averages is totally misleading. There is so much heterogeneity, even within E. coli population, that is totally underappreciated so far. These are very rich data sets.
But it seems that you could explain I mean, the data that you showed, you seem to be able to explain without really talking about the heterogeneity.
Yeah, so in this particular species that we discussed today, Vibrio alginoticus, there is probably somewhat less phenotypic heterogeneity. So we have some suspicions of some effects. Let me bring up one graph in particular. So here, for example, we see that for swimming segments at very high speeds, the two strains, phenotypes, or whatever, do behave quite similarly again, whereas we see the largest differences here. That's sort of the average behavior. And we suspect that what is actually happening is that within the blue population, we may have individuals with different levels of lateral flagella.
Right.
And the ones that are here might be the ones that look more like this. But this is speculative, so we don't know that for sure. In general, when we look at behavioral parameters, if we have species that only have a single polar flagellum, for example, we see a lot less variability in between individuals than for species like E. coli, where there's so much variation in flagellar architecture.
That's because the variability comes from how many flagella they've got. More or less.
Some of the variability, at least, yes.
Oh, thanks. That's
yeah, there are, of course, orthogonal axes of variability, like in internal signaling things, for example, but
oh, thanks.
Thank you. Any other question from the audience? Apparently not much at the moment. So first of all, I will